Okay, so I will start now. And today I will give a talk about genomic tracing reveals key genetic and epidemiological events associated with the early transmission of the SARS-CoV-2. And here is the contents of the topic. And first, I will show you integrated service platform of epidemiological and omics information we developed since the last year, just after the spread of COVID-19. And here we developed we developed an virus genome toolkit for SARS-CoV-2, and this platform has been online since last year, February 8th. It uses virus genome sequences and basic data resource, and various sample metadata and global epidemic statistics data, and also epidemiological data and virus research literature data. And our VERDTK systems continue to provide users with a high quality data set, as well as analysis tools that integrate genomics, epidemiological research literature, and other data from domestic and all world. And up to now, the VERDTK has collected a total of more than 4 million genome sequences of SARS-CoV-2, and after quality control processes, and our system can provide more than 4 million high quality sequence data for the SARS-CoV-2 genome and for the scientific research. And here we can also provide some daily report for the SARS-CoV-2 mutation information based on continuous growth of the meaning of SARS-CoV-2 genome sequence data. So we can give information for the mutation information and give a daily report. And here, so our whole systems consist of data warehouse and also based on some data standard, and we can also provide some genome annotation pipeline for the scientific research support. And here, so we can give you a lot of information related with SARS-CoV-2 for the mutation, for the evolution trend, and also some knowledge graph related with SARS-CoV-2. And here, so we can also give you some development for the outbreak of the major contents here. And here, so we also develop some pipelines can evaluate some special mutations for SARS-CoV-2, and whether or not this kind of certain mutations will be induce some damage for the vaccine, and also can evaluate whether or not these special mutations from the SARS-CoV-2 genomes maybe bring some risks. And we in this year we also evaluate some B.1.617.2 this India cases in the emerging China, and what kind of damage will be emergent. And so, and we give some annotation based on our pipelines, and actually, so we found our prediction for this kind of potential damage induced by some mutations, and our evaluation is quite right now. And here, so we want to show you our recently work about the molecular tracing model based on the topological segmentation algorithms and a knowledge graph. And I'd like to start with the background and problems. And we know the problem of SARS-CoV-2 molecular tracing is to make a phylogenetic chain between the same type of the rapidly evolving species that does not have a distant relationship cross species. And for the sequence of the closely related or the homological species, the maximum parsimony method is usually used. And the maximum parsimony method does not require the selection of a base substitution model. And the problem with the maximum parsimony method is that it is impossible to judge whether the finally constructed tree is the optimum result. And although there are two elements for model selections, one element is the model of the substitution between bases, and the other element is the model of the relative rate of all the substitutions at different positions in the sequence. In fact, there is another question. If a recombination occurs, how should the model be described? Okay, and the point 4 is the distance-based phylogenetic tree uses distance to cluster various strains. While strains with a shorter distance cluster more closely than those with a longer distance. The advantage of this method is that it is fast and flexible. The disadvantage is that the generated tree can be changed according to the algorithms we choose, and it cannot be tested to ensure that we are using the better model. So the tree may be inaccurate and unstable. And because the distance between various strains is calculated in pairs, this means the binary tree, and then pushed to the whole field to construct the phylogenetic tree, is a method that starts from the local optimum and finally obtain the global phylogenetic tree. Actually, the local optimum does not guarantee the global optimum, and whether it is a local optimum depends on whether it is guaranteed by the substitution model. If the base substitution model is incorrect, if the combination occurs, it may be difficult to achieve the local optimum, but below the global optimum. Okay, what is the solution we should try to have? Because we don't know what kind of the base substitution model is suitable model at the beginning of the tree. And the second, if there is a recombination between the viruses, in theory, if two or more virus strains infect the host at the same time, recombination may occur. How to establish an algorithm that best fit the true spread scenario of the virus, carry out the molecular tracing of the virus back, tracking and tracing the spread path of the virus. Can it be considered? Here, the first point is, this is a reverse, maybe a reverse engineering problem. Suppose that the virus transmission process is a process that leads to environmental adaptation through dynamic programming, and finally achieved the best transmission effect, that is a process that starts from local optimization and finally reaches global optimization. The second point is then our model should start from the actual real scenario. The virus transmission is not a binary tree. Okay, so what characteristics should we develop that new algorithms for the molecular tracing? The first is it should fit the real spreading scenario of the virus. It should be different from the traditional method of predicting the spread path of virus based on a certain base substitution model and the binary tree method that commonly used in virus molecular tracing. And the third, adopt reverse engineering strategy similar to the dynamic programming model of the BLAST. Temporarily, it's no longer necessary to consider the molecular clock hypothesis, base substitution model, and the recombination methods. Okay, so for propose our new molecular tracing algorithms, first we use an parsimony informative set, PIS, as the starting point for calculating the distance between virus strains. By constructing a Hamming distance matrix, SDM, weighted with a set conservation, there is no need to consider one. What kind of the base substitution model is suitable model? And second, whether recombination occurs between viruses. The second starts from any virus strain through the weighted Hamming distance matrix is WHGM, obtain the global virus transmission phylogenetic network map. Regardless, obtain the global virus transmission phylogenetic network map as a two-dimensional topological space, and then according to the local hardness of the two-dimensional topological space, segmenting the topological space topologically by using certain machine learning methods. And then so we can obtain the topological segmentation diagram of the phylogenetic network map, composed of the local classes containing the spatial temporal information of the virus transmissions. Adjust the global conservation frequency mu of the parsimony information set, and then change the mu to find a region. When mu changes in this region, the topological structures of the topological segmentation map remain unchanged, and the certain conservative frequency mu C is selected in this region as a cutoff. And then so the invariance of the transformation ensures that the virus transmission systems has a small potential energy of the network system and is in the global optimal state. Okay, then at the connecting line between or among topological class reveals the path information of the virus spatial temporary transmission, which can be used as the basis for molecular tracing of the virus spread. It's some steps we performed based on our proposed algorithms. We can start the molecular tracing. And first, establishing the phylogenetic network of the virus genome system based on parsimony informative set, PIS, and perform topological transformation by dividing the clustering phylogenetic network according to its topological structure SKLRD, and combining the knowledge graph formed by the epidemiological data, establishing the relationship between the topological associated chain and the macro transmission chain, and try to analyze the evolution patterns in two scenarios of the time and the space, and then use traditional molecular evolution analysis must combine with the specific epidemiological information of the key sequences samples to verify our math and the results. And here is a step-by-step. We perform our algorithm. At first, we collecting the high quality SARS-CoV-2 genome sequencing data from all over the world. And then starting from the parsimony informative site, PIS, based on the neighbor joint mask, introducing the PIS mutation frequency to weight the Hamming distance matrix, and then performing the topological morphism transformation to obtain a two-dimensional topological space, connect the two tuples in this two-dimensional topological space to construct a phylogenetic network map for virus transmission, and divide the topological space into subspaces according to the certain rules, and the topological subspaces have their own basic PIS models. And finally, analyze the topological space, clustering the virus evolution group, and further identifies the main mutation set and the main spread paths. This is some detail about algorithms, and here is the quality control for the obtained genomic data, and also calculation PIS, and also perform the two-dimensional plane mapping, and also construction of the collection map for the molecular treatment, and also perform topological segmentation for our phylogenetic network map. And then determine the invariance area of the topological segmentation, and transformation are based on the conservativeness of the PIS frequency weighted factor nu, since the granularity of the system's molecular variation map will vary with the change of the frequency weighting factor nu. You can observe the structural changes after the derived map of the topological segmentation by adjusting the frequency weighting factor nu. In this case, nu changes within a certain range. The topological structure of the segmentation map remain basically unchanged. And then, so this means here we got the point here, the lowest potential energy position, and the system is in the optimal state. Then we can also divide our phylogenetic network map into individual topological class, and the line among the individual topological class is indicate the spread path of SARS-CoV-2 around the world. This is finally based on our algorithms. We can give a very detailed spread path of SARS-CoV-2 and around the world. And together with the knowledge graph of SARS-CoV-2, and this knowledge graph, so we constructed it by collecting the information from the literature, the newspaper, from news agency around the whole world, and from 167 countries. Then so put this epidemiological information together with our topological segmentation algorithms. Then so we can perform very accurately molecular tracing. And here, so we also perform some confirmation to our algorithms. We using some famous study results. I try to use our algorithms to verify the results. And for example, this is published on the Science in 2004. Is by Professor Guoping Zhao, and they worked in for tracing the SARS origin, and we collected the data from this article and put in our calculation systems. We get the totally similar result, and although we also get the data from some recent article published on the VENAS, and we get all of the exactly the same results. That means our algorithms work quite well. And this is some pictures we can see. This is individual topological cloud, and from this cloud, and you can clearly see spreading paths of SARS-CoV-2 around the whole world from this picture, and it's really very clearly we can give some reasonable spreading paths for SARS-CoV-2. This is all some results based on our calculations. And here, so we would like to show you some phenomena we discovered by our calculations. We found some interesting phenomena. That means for the temporal spatial spreading of the SARS-CoV-2, there exists a 4-stage fluctuation phenomena for the early spread SARS-CoV-2 around the whole world. Yeah, and our analysis shows that the emergence of the SARS-CoV-2 from its outbreak to the global spread at the early stage shown this interesting phenomena. For example, from local outbreak for early spread, and we can find a very exact evidence for the genetic drift, for the founder effect, and also a parallel evolution, and the large-scale spread, and the recurrent mutation, and the global spread, and the convergent and close adaptation. And although the whole evolution process can be divided into 4 stages in time, the 4th stage, the relatively concentrated outbreak in the early stage from 2019 and December 24, and it's almost 2 years ago, and to the 2020 and January 18, and the multi-point and multi-source outbreak have accumulated a high potential energy of virus genetic diversity, which is conducive to the spread of the virus to many places around the world. The second stage is early, relatively concentrated outbreak from 2020, from January 19 to 2020 and March 15, the middle of March. And in the process of the global regional spread, genetic drift occurs, and the genetic diversity of the virus is largely lost in the region, and the regional evolution of the SARS-CoV-2 presents the characteristics of the founder effect. And the parallel evolution and the adaptability of the SARS-CoV-2 to human hosts shows the regional, environmental, and individual selectivity. The third stage is global spread in the middle and later stage, from the middle March 2020 to the end of April 2020. In the process of the global spread of the SARS-CoV-2, as countries have variable prevention and control at the beginning of the stage, the flow of the people and the number of the cases have increased rapidly. And the viral genetic loss of the diversity and the diversity restoration repeatedly intervened, and the current mutations occurs, mutation frequency is unstable, and parallel evolution transition to the convergent evolution, and the founder effect gradually disappear. And the fourth stage, we would like to say, the stage for the global spread of SARS-CoV-2. From the beginning of the May last year and August to the August of the last year. In the later stage of the global spread of the SARS-CoV-2, various virus strains are fully mixed. The genetic diversity, recovery, parallel evolution, convergent evolution, founder effect, and the recurrent mutations of the SARS-CoV-2 have basically disappeared. The mutation frequency is relatively fixed, and the SARS-CoV-2 has gradually adapted to the human host. During this period, the abundance of the genetic diversity of the SARS-CoV-2, defined by the sum of the polymorphisms of the main functional genes of SARS-CoV-2 and the population distribution, has greatly increased, and the lethality rate has continually maybe to decline, indicating that the SARS-CoV-2 is gradually adapting to the human host. It's maybe a current phenomenon. Okay, this is what we discovered by our calculations. And now I would like to give a summary. And first, we introduced a knowledge enhancement and topological reconstruction strategies to improve the traditional phylogenetic network math and establish a new phylogenetic network topological analysis math. And the feature of the new math is that start from the constrained average analysis of the big data to eliminate the uncertainty caused by a large amount of data and summarize the epidemic trend of the disease, and conveniently discover the evolutionary pattern of the SARS-CoV-2 in the process of global spread in accordance with the law of evolution. And through the topological segmentation of phylogenetic network, the intuitive, even fragmented evolution mechanisms of the SARS-CoV-2 in the spread chain of different regions of the world can be spliced and integrated, deducing the possible evolution mechanisms of the SARS-CoV-2 from the outbreak to the global spread. And in the fourth hypothesis of our discovery, it can be noted that the first to the fourth stage from the closed loop, according to our hypothesis, the global spread of the SARS-CoV-2 is actually a wave fluctuation process. But when and where does the fluctuations processes before the outbreak in Wuhan begin to happen? Data and calculation support are still needed. And simply, the time span from the outbreak in Wuhan to the current epidemic is almost 2 years. According to our hypothesis, before the outbreak in Wuhan, SARS-CoV-2 may have been spreading and evolving, went through the same fluctuation process, and because the virus evolution before the outbreak in Wuhan, like human interference, and it is in the state of the neutral evolution, the fluctuation cycle period of asymptomatic infection, and its periods may have lasted for several months, a year, or even longer. This is our conclusion by the calculations based on our algorithms and the discovery. And the data generated by the biological experiments contain the laws of biological behind it. The real biological signals contained in the data can form the specific topological structure in the specific topological space. And the formation of this topological structure follows specific mathematical principles. Science structure determines function. Starting from the specific topological structures, it can explain or discover basic biological mechanisms, and biological obeying specific mathematical principles in specific mathematical species. This is the first point is some consideration result from our algorithms dealing with the genomic data of SARS-CoV-2. The last point is some consideration, maybe to tell, it can tell us what we can use in some basic mathematical math and rules to dealing with biological data to have some interesting discovery related with the biological function. Okay, thanks for the listening. It is my talk today.
Okay, is there any question?
It's maybe the mathematical is a little bit difficult to understand. So, okay, so yeah, questions.
Thank you very much for your very lucid and very good presentation. It was really very educating and very knowledgeable. I have two short questions. Number one, that how the concept of potential energy in the cluster is associated with the emergence of the new highly transmissible variants. And my second question is that, do you think that genetic drift is associated with the loss of diversity of viruses more than the emergence of more infectious strains, which basically wipe out the genetic diversity due to higher transmissibility? Thank you very much.
Excuse me. So because maybe some problems I cannot clearly listen your questions. Can you little bit repeat it again? Maybe you can make a distance from your microphone, and because and sorry, so the voice is a little bit not very clear. So can you repeat your question again?
My first question is that how the emergence of new variants like Omicron and Delta is associated with your concept of potential energy in your phylogenetic clusters.
I really cannot get your point. And who can give some clearly description about his questions? And maybe Kajabri, so Professor Kajabri.
Yes, Dr. Yusili, I think she's asking new variants, particularly strongly infectious strain called Omicron. How you explain emergency of Omicron, particularly in relationship to particular gene or something? I think this is her first question.
Okay, so this question is not related with our calculation, but I would to say, so actually 2 weeks ago, CDC of China, so they sent some message to me to ask whether or not the Omicron will bring a high risk to the human beings now. But I think so maybe Professor Kajabri have the similar consideration as that. So it's really very strange in the spec proteins. They have more than 30 some new mutations. It's really unusual. So normally for some species they have this kind of randomly very huge of the mutations. Normally it is not good for the species themselves. And then so I guess maybe this kind of mutations in the genomes maybe in the most of the cases will damage the survival ability of the viruses, and then so maybe it will not be very dangerous for human. Maybe it's maybe some bring the minor risks for the human. And recently, you can see it is maybe true. Even the damage and bring the risk is lower than the Delta strains. It is somehow reported by the recently the evidence. So, yeah, this is my opinion.
Okay. So maybe was the answer enough? I'm just gonna read what was typed in the chatting screen. How your concept of potential energy in the phylogenetic tree can explain the emergence of highly infectious variants. Do you think that this is genetic drift, which is associated with the loss of genetic diversity and not the highly infectious variants, which wipe out less transmissible variants? Oh, that's very long question. So
it's okay. So yeah. Okay, so at first I would like to say why so we should select some cutoff for the conservative parameter. The new, it is some conservative score for the site conservativeness for the parsimony informative set. And actually, so when we put this phylogenetic network map on the two-dimensional topological space, and actually, because we get the data from the real scenario, and we consider the final network is already optimum globally. Then, so we try to divide this whole picture into individual topological class. And then so we repeat the every step how we construct the final map. Then so by step reconstruct the final map. So we change the score of the selected cutoff for the weighted parameter new, and then so we can find the structure of the individual topological class for the whole pictures will be changed by changing the cutoff mu. And if we find in some region, we new in this region, the score in region, and the structure, the whole map is unchanged. And here, indirectly means we reach the smallest potential point, and then so we get the final map is stable and is can be used to perform molecular treatment. And this is normally for the bioinformatics study. So we should consider the factors, and we should build some systems, and we should try to find whether or not this systems is in the stage of the smallest potential energy. Only in this stage we can give some results is the most related to the real and maybe biological mechanisms. And almost for our bioinformatics studies, so not so many people consider, make some consideration for this point. Yeah, this is my answer. Yeah.
Okay. I have one more question, and can you maybe answer in 30 seconds? The question was, are we expecting another more contagious or more infectious variants? What could be the possible reason for the spike sequence dynamics? Could you maybe answer in 30 seconds? Maybe?
Sequence dynamics. Okay, so, and actually, if you change the conservative score, this the parameter is new, is frequency, and then so you will select a certain number of genome data from the whole database. And then that means you can perform this phylogenetic network map dynamically, and you can also step by step to perform this reconstruction of the whole map, and by using this cutoff number, change the cutoff number, and you can perform, I would like to say, the constraint technology, and to find the optimal data number for the molecular tracing results. This is maybe dynamic selections for the citizen data. So this kind of rich scale of the data size is suitable for perform the molecular tracing. That can be by using this kind of method to get the best results.
Okay, thank you, Dr. Lee, and thank you for the questions from our Pakistan colleagues from ICCBS.